Welcome to the Deep Dive, where we take a stack of complex sources, distill the core knowledge, and give you the fast track to expertise. Today, we're taking a microscopic look at a really fundamental way our tissues respond. Responding to demands, stress, or injury by,
Right.
well, by aggressively multiplying their numbers.
That's right. Our focus today is hyperplasia, a concept that sits right at the heart of pathology, but honestly, also at the core of life itself.
Mhm.
If we want to understand adaptation, regeneration, and ultimately how things like cancer get started,
Mm.
we really have to get our heads around why and how cells make more of themselves.
Okay, let's try and unpack this with a nice, clean definition first.
Yeah.
Our sources are, well, they're extremely clear here. Hyperplasia is defined as an increase in the number of cells within an organ or tissue.
Yeah.
It seems to be a very highly ordered, organized response to some kind of specific trigger.
And it's important, I think, we spend a moment clarifying that because it's easily confused, isn't it, with its sort of structural sibling, hypertrophy. Hypertrophy is the increase in cell size. The cells themselves swell up, get bigger, but crucially, no new cells are actually added.
Ah, okay. Like lifting weights.
Exactly like that. Think of the skeletal muscles of an athlete. They get bigger, the muscle fibers swell, but the total number of muscle fibers generally stays fixed. That's hypertrophy.
Got it. So that distinction is really vital for understanding disease then. Hyperplasia, where you get more cells, well, our sources say it can only happen in tissues where the cells can still divide.
Or, and this is critical, or if they contain a good reservoir of tissue stem cells.
Okay.
Tissues that can't divide, like most heart muscle cells or neurons in the brain, they pretty much can only respond by getting bigger, by hypertrophy.
Right. And what's interesting, I found, is that in many of these dynamic tissues, the ones constantly renewing or adapting, like maybe the breast or the liver or adrenal glands, these two processes, hyperplasia and hypertrophy, they often happen together.
Oh, absolutely. They often coexist. The tissue gets larger overall because the cells are both swelling up. That's the hypertrophy bit, and they're multiplying. The hyperplasia bit. It's like the body's way of maximizing the total functional capacity. Double whammy.
So our mission today then is to dive deep into the triggers, the hormones, the growth factors that kind of orchestrate this rapid increase in cell count.
Yeah.
And then, maybe more importantly, we have to spend some real time understanding the incredibly thin sort of molecular line where this necessary adaptive survival thing, transforms into a problem, into a dysregulated state that could even be fertile ground for malignancy later on.
It's a fascinating balance, isn't it? Hyperplasia is really a testament to the body's just phenomenal capacity for controlled growth. But the very mechanisms that regulate this control, they're the same ones that, if they get tipped just slightly off balance, can initiate that unchecked proliferation we worry about.
Okay, let's dig into the details then. Okay, so we established the sort of dividing rule. The tissue needs cells capable of division. But let's refine that a bit. What does capable of division actually mean, like down at the microscopic level?
Well, it relates directly to how we classify cell populations in the body. Tissues get categorized based on their inherent ability to enter the cell cycle, to divide.
Right.
So you have what we call permanent cells. Think neurons, heart muscle cells mainly. They're terminally differentiated. They've reached their final form and function, and typically they just can't divide anymore.
Okay, fixed population there.
Then you have stable cells.
Yep.
Liver cells, hepatocytes are a great example. They're normally just sort of resting, quiescent in what we call the G0 phase of the cell cycle. But, and this is key, they can be rapidly stimulated to jump back into the cycle and start dividing if needed.
Okay, so they're on standby. And the third type?
The labile cells, exactly. Labile cells, like the epithelial cells lining your gut or your skin, or the cells churning away in your bone marrow, they're just constantly dividing and replacing themselves throughout life. It's their job. So hyperplasia is naturally most impressive and often fastest in these labile and stable cell populations because, well, they've got the molecular machinery ready and waiting to respond when that signal comes.
And that signal, the trigger you mentioned, it's non-negotiable. Hyperplasia isn't just a random event.
Never. Absolutely never. It's always a specific targeted response to a particular stimulus. Now, if the stimulus is normal and necessary, like, say, the hormonal rush during pregnancy or the need to replace tissue after surgery, we call that physiologic hyperplasia. It's normal adaptation.
Okay, the good kind.
The good kind, yeah. But if the stimulus is inappropriate or excessive, or maybe it just goes on for too long because something's gone wrong internally or externally, then we call it pathologic hyperplasia. Now, importantly, even though it's pathologic, The process itself is often still technically controlled by that ongoing stimulus. It's just that the stimulus itself is wrong, and that's the one that raises the alarms.
Let's start with the good kind then, the physiologic stuff, the adaptation that keeps us going.
Yeah, physiologic hyperplasia is really adaptation at its best. It happens when there's a clear, calculated need in the body. Either you need to ramp up the functional capacity of an organ that responds to hormones, or you need a rapid, large-scale compensation after some tissue has been damaged or removed, like in surgery.
Okay, the classic examples seem rooted in hormonal signals, right? Thinking about like sexual development, reproduction. Let's talk about the mammary glands.
Oh, the female breast is just a masterclass in controlled hyperplasia. At puberty, and then much, much more dramatically during pregnancy, the glandular epithelium, the milk-producing parts, undergoes this massive proliferation.
And that's purely hormone-driven.
Entirely driven by a sort of sequential flood of hormones. You get estrogen, which initially drives the ducts to proliferate. Then progesterone comes in, stimulating the little sacs, the alveoli, to differentiate. And finally, prolactin, which gets the machinery ready for actual milk production.
Wow. So the body is rapidly just expanding the number of these glandular units, getting ready for a future function. And I guess it's not just adding cells randomly. It sounds like the whole architecture expands in a really organized way.
Oh, completely. It suggests a really complex, synchronized orchestra of growth factors, all coordinating that response.
Yeah.
It is a remarkable orchestration.
Yeah.
And like we mentioned earlier, this is exactly where hyperplasia and hypertrophy work together beautifully. Those newly added glandular cells, they're simultaneously enlarging, that's the hypertrophy, to maximize their potential output. It's like a system-wide controlled command, maximize production capacity now.
Makes sense. But maybe the most dramatic example, certainly the fastest type of physiologic hyperplasia, comes under that other category you mentioned, compensatory regeneration, specifically the liver.
The liver. Its regenerative capacity is, frankly, legendary in biology. After a partial hepatectomy, where surgeons might need to remove, say, up to 70% of the liver, perhaps for a living donor transplant.
Sure.
The remaining tissue just rapidly proliferates until the original mass is essentially restored. It's probably one of the clearest examples we have of growth factor signaling, just forcing those normally resting, stable cells, the hepatocytes, out of that G0 phase and straight into division. Boom.
So if I understood the sources correctly, it's not like the missing lobe magically reappears.
No.
It's more that the remaining tissue just expands its existing structure, fills the space until the functional mass is back.
Precisely that. And the scale of it is immense. We're talking substantial regrowth within days. The primary driver here seems to be the release of specific growth factors, notably things called hepatocyte growth factor, HGF, and transforming growth factor alpha, TGF-alpha. These molecules basically act like keys, locking onto receptors on the surviving liver cells. And that activates these intricate signaling cascades inside the cell that just propel them into the cycle of division.
Okay, that makes perfect sense for a solid organ like the liver. But what about a system that's already constantly turning over, like the bone marrow? What happens there if, say, there's acute blood loss? The body needs a huge, immediate increase in production, right?
Right. And the bone marrow demonstrates probably the most impressive acute acceleration of hyperplasia we know of. If someone suffers massive bleeding or maybe an episode of severe hemolysis where red blood cells are destroyed, the demand for new red blood cells just skyrockets instantly.
How does the body, tell the bone marrow factories about that urgent need?
Primarily, it's the kidney acting as the sensor. Low oxygen levels in the blood, called hypoxia, trigger the kidney to release vast quantities of a hormone, or actually, it's better described as a growth factor called erythropoietin, EPO.
EPO, right.
EPO is essentially the chemical commander that orders massive hyperplasia, specifically in the erythroid lineage, the red blood cell precursors within the bone marrow.
And we're not just talking about, a small boost, a 50% increase in production.
Oh, not at all. The source material really highlights this. The bone marrow's red blood cell production capacity can ramp up by as much as 8-fold in a really short period.
8 times? Wow.
Yeah. This massive proliferative response, orchestrated almost entirely by EPO stimulating those progenitor cells, ensures the body can recover relatively quickly from potentially life-threatening blood cell deficiencies. And the beauty of this system is the speed, but also the feedback loop. Meaning, once the number of mature red blood cells in circulation gets back to normal, the oxygen levels rise, the kidney stops pumping out so much EPO, and the hyperplasia in the marrow shuts down. It returns to baseline. That's the very definition of controlled adaptive necessity. It turns on strong, turns off when done.
Okay, that eightfold increase in the bone marrow. It really shows how incredibly powerful that growth factor machinery is, which I guess leads us to the really interesting bit. How does the system know the difference between a temporary acute need like that blood loss and the signal that's sustained, inappropriate, maybe even dangerous? That distinction brings us to pathologic hyperplasia.
Exactly. Pathologic hyperplasia generally results when those same powerful growth factors or hormones are acting in a way that's excessive or goes on for too long or is just plain inappropriate for the situation. The cell's machinery, it's still being obedient, it's following orders.
Right.
But the signal it's obeying is faulty. Now, crucially, and this is a really important distinction we'll come back to, even though it's abnormal, the growth remains dependent on that stimulus. If you can somehow remove the signal, the hyperplasia typically regresses. It's still under external control, albeit faulty control.
Okay. Let's look at maybe the classic example of hormonal excess, endometrial hyperplasia. This seems to link right back to the healthy hormonal responses we were just talking about in the menstrual cycle.
It does, perfectly. In a normal cycle, the hormone estrogen drives the proliferation of the endometrial lining, that's the hyperplasia part, in the first half of the cycle. It's preparing the uterus, potentially, for an embryo to implant.
Okay.
Then in the second half of the cycle, after ovulation, progesterone is released. And progesterone acts as the sort of anti-proliferative brake. It tells the cells to stop dividing so much and start differentiating, maturing, limiting further growth.
So what happens when that balance, the estrogen driving growth, the progesterone stopping it, gets broken?
Well, if you get a disturbance that results in an absolute increase in estrogen, maybe from certain ovarian conditions or tumors, or actually more commonly, a relative increase in estrogen because there's not enough progesterone to balance it out.
Ah, like if ovulation doesn't happen reliably?
Precisely, like in an ovulatory cycles. In that situation, the estrogen keeps stimulating proliferation, but the progesterone brake never gets applied properly. So the endometrial glands just keep proliferating excessively.
And what are the clinical consequences of that excessive growth? I mean, besides the obvious one, which is often abnormal uterine bleeding.
Right. Abnormal bleeding is the hallmark symptom. But beyond that, this is where the morphology, what the tissue actually looks like under the microscope, starts to matter a lot. And it's where the risk factor really emerges.
How so?
Endometrial hyperplasia isn't just one single thing. Pathologists categorize it based on severity and, critically, on whether the cells look normal or abnormal. We talk about simple hyperplasia, complex hyperplasia, and the one that really worries us, atypical hyperplasia.
Okay, what does that atypical label mean? What's different?
Atypical hyperplasia means the cells aren't just multiplying too fast. They're also starting to show abnormal changes in their appearance, their shape, their nuclear features, how they're arranged. Basically, they're showing signs of dysplasia.
Dysplasia, like pre-cancerous changes.
Essentially, yes. This atypical growth pattern has by far the highest likelihood of progressing over time to become a full-blown endometrial cancer, an adenocarcinoma. The thinking is that the excessive hormonal stimulus has been going on for so long, forcing so many extra rounds of cell division, that the probability of accumulating critical DNA mutations, those genetic errors we talked about, has become alarmingly high in those cells.
Wow. That really deepens the understanding of the pathology, doesn't it? It's not just about having too much tissue. It's about the quality control breaking down because of the sheer speed and duration of the growth.
Exactly right. Now let's shift gears slightly and look at benign prostatic hyperplasia, BPH. This affects, well, a huge number of aging men. It's another classic study in hormonal imbalance, but this time involving male hormones, androgens.
We often talk about androgens generally, but specifically, which molecule is actually driving the proliferation, the hyperplasia in the prostate gland?
It's primarily a molecule called dihydrotestosterone, or DHT. Testosterone, which is mostly made in the testes, circulates in the blood. But within the prostate gland itself, specifically in the supportive stromal cells, it gets converted into the much more potent androgen, DHT.
By an enzyme.
By an enzyme called 5-alpha reductase. And it's this DHT that then acts powerfully on both the stromal cells and the epithelial cells of the prostate, stimulating both hyperplasia, more cells, and hypertrophy, bigger cells. And that leads to the gradual, often problematic, enlargement of the gland over time.
So in this case, the inappropriate stimulus isn't necessarily a big systemic storm of hormones like the estrogen issue.
Not usually, no.
It's more of a localized metabolic conversion process that just becomes sort of exaggerated or more effective with aging, and it's sustained over decades.
That's a great way to put it. And, that understanding has had massive therapeutic consequences. Because we know BPH is driven by DHT stimulation, we now have excellent medications, the 5-alpha reductase inhibitors you hear about.
Finasteride, dutasteride.
Exactly those. They work by blocking that enzyme, blocking the conversion of testosterone to DHT right inside the prostate. You effectively remove the primary growth stimulus, and often the hyperplasia regresses, prostate shrinks, and symptoms improve.
Which perfectly demonstrates that critical point you made earlier. Pathologic hyperplasia, even BPH, generally remains controlled by and reversible if you can remove that driving stimulus.
Precisely, unlike cancer, which we'll get to.
Okay, and finally, let's touch on external hijackers. Virally induced hyperplasia. This sounds like it takes the concept of a faulty internal signal and turns it into more of a full-scale hostile takeover by an outsider.
Yeah, this is perhaps the most direct and worrying route from simple hyperplasia towards malignancy. Certain viruses, most notoriously the human papillomaviruses, or HPV.
The ones that cause warts.
Exactly. They cause infections that result in things like common skin warts or, more concerningly, mucosal lesions, for example, on the cervix. What you're seeing there are massive, localized hyperplastic growths of the epithelium, just tons of extra cells piled up.
How does a tiny virus actually force my cells to multiply against my body's own control systems? That seems incredible.
It's a truly genius, albeit quite sinister, molecular strategy. The virus encodes and introduces its own proteins, its own factors that are specifically designed to target and neutralize the host cell's most important regulatory proteins, the ones we call tumor suppressors.
Okay.
For instance, the high-risk HPV types, the ones linked to cancer, produce two particularly nasty proteins known as E6 and E7.
E6 and E7, what do they do? What do they attack inside the cell?
Well, E7 specifically targets and promotes the degradation of the retinoblastoma protein, RB. Now, RB is basically the key gatekeeper molecule that normally prevents a cell from entering the S phase, the DNA replication phase, of the cell cycle, unless conditions are right. E7 takes out that brake.
Wow. Okay, so the brake is gone. What about E6?
E6, acting perhaps even more sinisterly, targets and promotes the destruction of the p53 protein. p53 is often called the guardian of the genome. It's the cell's chief sensor of DNA damage and stress, and its job is to halt the cell cycle for repairs, or, if the damage is too severe, to apoptosis program cell death. E6 takes out the guardian and the emergency kill switch.
So the virus knocks out both the main brake and the emergency stop button, p53.
That's essentially it. By neutralizing both, the virus effectively forces the host cell into uncontrolled division. Why? Because it needs the host cell's replication machinery to copy its own viral genome. It hijacks the factory.
That's terrifyingly efficient. It's not just, turning up the volume on growth factors from the outside. It's actively dismantling the internal checks and balances that are designed to prevent exactly this kind of aberrant growth.
Precisely. And the result is this frantic, forced cell division, leading to those massive hyperplastic lesions, the warts or cervical lesions, and crucially because the main control systems, especially p53, are gone or disabled. Those rapidly dividing cells are now highly prone to accumulating other random DNA mutations, the secondary hits that are necessary for the final transition from just hyperplasia to invasive cancer. The virus creates the perfect storm.
Okay, we've talked a lot about the signal, the growth factor, the hormone, maybe the viral protein acting like a signal. Now we need to sort of get inside the cell and watch the engine actually turn over. If hyperplasia is fundamentally about cells dividing, we need to understand the molecular instructions that force a cell to ignore its resting state and start replicating its DNA and splitting in two.
Right, we're moving from the sort of tissue level down to the nanoscale, the molecular pathways. Hyperplasia ultimately is driven by the activation of two main kinds of mechanisms, and often they act sequentially or sometimes together.
What are they?
First, you can stimulate the existing mature cells to start dividing again. That's proliferation. And second, particularly in tissues capable of it, you can activate the resident tissue stem cells to turn out more new cells. That's more like regeneration from a deeper source.
Okay, let's focus on the first one. Proliferation stimulating those mature, maybe resting cells. For a cell to divide, it has to get past that critical checkpoint in G1, the restriction point, and commit to entering the S phase, where it copies its DNA. How does a growth factor, like that HGF in the liver or EPO in the marrow, actually initiate that sequence?
It all begins at the cell surface with receptor binding. The growth factor molecule acts like a specific key fitting into a highly specific lock, its receptor protein embedded in the cell membrane.
Okay.
This binding event, this docking, triggers an immediate chain reaction inside the cell. It's usually a cascade of biochemical events, often involving adding phosphate groups to proteins, phosphorylation that relays the signal inwards. We call this a signaling pathway.
Right. Are there key signaling pathways that are consistently involved when growth factors are driving proliferation?
Yes, the sources point primarily to two major interconnected pathways that are really central to this. The first one is often called the MAPK pathway. That stands for mitogen-activated protein kinase.
MAPK pathway, okay.
Activation of this pathway essentially relays that grow and divide signal all the way from the cell membrane receptor through a series of kinases in the cytoplasm right into the nucleus.
And in the nucleus.
In the nucleus, it activates specific proteins called transcription factors. And these transcription factors then turn on the genes that are absolutely essential for the cell to prepare for and execute cell division. Things needed to copy DNA, build new membranes, etc.
So MAPK is like the primary divide now instruction pathway. What's the second major one you mentioned?
The second one is the PI3K ACT pathway. Now, while MAPK is maybe heavily focused on driving the cell cycle progression, the PI3K ACT pathway is often thought of as being more about cell survival and growth and metabolism.
How does that contribute to division?
Well, when it's activated by growth factors, it does a couple of crucial things. It actively suppresses the cell's suicide program, apoptosis, making the cell more resistant to dying. And it also ramps up nutrient uptake and energy synthesis pathway.
Ah, so it provides the resources.
Exactly. It creates the metabolic resources and the survival signal necessary to undertake such a demanding process as copying your entire genome and dividing. You really need both the clear instruction to divide, MAPK, and the resources and survival support, PI3K ACT, to do it effectively.
So the growth factor hits the receptor, turns on the key, which activates MAPK, the division command, and PI3K ACT, the resource and survival supplier. And these work together to push that cell past the G1 checkpoint and into the cycle.
That's a really good summary of the core concept. Now, if we revisit that liver regeneration model we talked about, it shows how complex this can be in a real tissue. It's often not just one single signal hitting go. It can be a precisely timed sequence of events.
How is it sequential in the liver example?
Well, the evidence suggests that when a large chunk of the liver is removed, the remaining cells don't just immediately start dividing. First, they get sort of primed. There's an initial wave of signals, often involving inflammatory mediators released locally, cytokines like TNF, tumor necrosis factor, and IL-6, interleukin-6.
And these don't make the cells divide directly.
Apparently not. They don't seem to be strong mitogens themselves, but they make the hepatocytes responsive to the next wave of signals. They kind of prepare the cellular environment, get them ready.
You're setting the stage.
Exactly. Then the second phase kicks in. The true progression factors, like the HGF and TGF-alpha we mentioned, arrive and bind to those now primed receptors. These are the molecules that strongly activate the MAPK and PI3K pathways, really forcing the cells into mitosis.
Ah.
This kind of sequential process likely ensures a rapid but still controlled expansion that's also self-limiting. Once the liver reaches its optimal mass again, these signals presumably die down.
Right. But what if that first mechanism fails? What happens if those surviving mature liver cells are maybe too damaged, say from chronic hepatitis or cirrhosis, and they just can't respond properly to the growth factors to start dividing? This brings us to that stem cell fallback plan you mentioned earlier.
Yes. This is where the body often demonstrates its remarkable deep reservoir capacity for repair. If the proliferative capacity of the main workhorse cells, the differentiated hepatocytes, is compromised, the body appears to switch strategies. It activates a population of less differentiated cells that reside within the liver, often found near the bile ducts. In rodents, they're sometimes called oval cells. In humans, we generally refer to them simply as intrahepatic stem cells, or progenitor cells.
And these stem cells are usually just sitting there, quiet.
Precisely. They're normally quiescent, just waiting. But they can be activated by specific signals, potentially different mixes of growth factors or changes in the local tissue environment that coax them out of dormancy.
And what do they do once activated?
They start to divide and, crucially, differentiate. They can generate new functional hepatocytes and also cells to line the bile ducts, effectively rebuilding the damaged tissue from a more fundamental level. Now, this stem cell-driven mechanism is generally thought to be slower, perhaps less efficient than just having the mature cells divide.
But vital if the first option isn't available.
Absolutely vital for tissue repair when that primary mechanism of mature cell division fails. So, you see, the engine driving hyperplasia has this direct drive mature cell proliferation, powered by MAPK and PI3K, and also this reserve gear stem cell activation and differentiation, all ultimately controlled by growth factor pathways that are immensely powerful and need to be highly synchronized.
Which brings us right back to the inherent danger, doesn't it? If these incredibly powerful growth-driving systems are activated by a sustained pathological signal, too much hormone, a virus, the sheer number of extra cell divisions required to maintain that abnormal growth dramatically increases the chance for mistakes, for systemic error.
Hashtag advancements and implications. The tipping point. This really does feel like the tipping point of the whole discussion, doesn't it? We've established that hyperplasia, even pathologic hyperplasia, is technically controlled.
Right, by the stimulant. But it dramatically increases the probability of error. Let's delve deeper into that connection. How exactly does pathological hyperplasia become what the sources call the fertile soil for cancer development?
It boils down fundamentally to probability and the fidelity of our DNA replication machinery. Our cells are remarkably good at copying DNA. Astonishingly accurate considering the scale.
3 billion base pairs.
3 billion base pairs copied every time a cell divides. But they're not absolutely perfect. Every single time a cell goes through that S phase, makes a complete copy of its genome, there's a small but finite chance of acquiring a random mistake, maybe a single base change, a point mutation, maybe deleting a small piece, maybe swapping bits around, a rearrangement.
Okay, errors happen.
Errors happen.
Yeah.
And the more often you force a cell population to replicate its genome, the higher the overall cumulative risk of one of those errors landing in a really bad spot.
It's just math then. More lottery tickets, more chances to hit the unwanted jackpot.
It's exponentially higher, really. Pathologic hyperplasia forces that cell population to undergo potentially dozens, maybe hundreds, of extra, biologically unnecessary rounds of division. And in that environment of rapid forced division, the cell often has less time for its DNA repair mechanisms to kick in and fix any mistakes before the next round of division starts. So this environment is just perfect for accumulating these genetic aberrations, these mutations.
So the hyperplasia itself, the excessive growth, isn't cancer.
Correct. It is not cancer.
But the process of repeated forced growth is what statistically creates the individual cells that might eventually become cancerous by picking up the right or wrong mutations.
That's exactly the concept. The first few mutations that occur might be completely harmless, maybe land in non-coding DNA, or don't change protein function. But eventually, purely by chance in that rapidly dividing pool, one of those random errors is likely to occur in a gene that's critical for controlling growth itself.
Like what kind of gene?
For example, a mutation that causes a proto-oncogene, like maybe the RAS gene, to become permanently switched on. That's a gain-of-function mutation. Or, perhaps even more commonly in many cancers, a mutation that disables both copies of a tumor suppressor gene, like P53 or RB, a loss-of-function mutation.
Okay. And once a cell in that hyperplastic tissue acquires one or more of those critical mutations, what happens to its relationship with the original external signal, its dependency on the excessive hormone or the growth factor that started the whole process?
That is the absolute defining shift. That's the molecular line between hyperplasia and true cancer. Pathologic hyperplasia, as we keep stressing, is characterized by its dependence on that external stimulus.
Right.
If you remove the excessive estrogen, or block the DHT, or maybe clear the HPV infection, the abnormal growth generally halts, and the tissue often regresses back towards normal. Why? Because the hyperplastic cells, even though they're overproliferating, are still fundamentally listening to and obeying those external commands.
But a fully malignant cancer cell, by its very definition, it's achieved independence, autonomy.
Precisely. Cancer cells have accumulated enough of these critical internal mutations. They've essentially hot-wired their own internal growth pathways, like activating RAS permanently, and silenced their own internal brakes, like deleting P53, so that they no longer need that initial external hormone or growth factor stimulus to keep dividing.
They're self-sufficient.
They've become self-sufficient regarding growth signals. They often ignore external inhibitory signals too, and sometimes even learn to produce their own growth factors. This transition from dependence on external trigger to autonomous, self-driven proliferation is the hallmark of malignancy, and it's driven by the genetic instability fostered within that preceding phase of sustained hyperplasia.
That makes the BPH treatment example even clearer. If BPH was already cancerous from the start, giving those 5 alpha reductase inhibitors to stop DHT production wouldn't work.
Exactly. It would have little to no effect.
Because the cancer cell is driving itself now, it doesn't care about the external DHT level anymore. The fact that the BPH does regress confirms it was still in that dependent hyperplastic state.
That's the clinical anchor right there. And of course, this understanding is what drives modern preventive strategies in situations like endometrial hyperplasia. If a biopsy shows not just hyperplasia, but atypical hyperplasia, that real fertile soil we talked about,
Yeah.
the recommendation isn't just watchful waiting. It's often aggressive intervention, maybe high-dose progestin therapy, to completely counteract and suppress the estrogenic stimulus, or sometimes even hysterectomy, to remove the tissue entirely. The goal is to force that hyperplastic tissue to regress before it accumulates those critical cancer-causing mutations, drastically lowering the risk.
Which means then that understanding those growth factor signaling pathways, we discussed the MAPK pathway, the PI3K pathway, it isn't just academic, it's actually the key to developing targeted cancer prevention strategies, isn't it?
Absolutely.
If we can find ways to specifically block the excessive signal early, maybe block the receptor, or inhibit a key kinase downstream, we could potentially prevent that whole dangerous environment of rapid division that creates the genetic mistakes in the first place.
It's the ultimate upstream target, isn't it? And we have seen incredible advancements in precisely these kinds of targeted therapies. Drugs designed to specifically inhibit growth factor receptors, like EGFR inhibitors, or downstream kinases in those signaling cascades.
Right.
These are becoming increasingly important not just for treating established cancers, but potentially for intervening in high-risk precancerous hyperplastic conditions to stop them from ever achieving that autonomy.
It really is fascinating. It turns out that a cell's seemingly simple adaptive mechanism, just making more copies of itself when needed, is somehow both the life-saving engine behind regeneration and healing, and simultaneously the body's first, most dangerous vulnerability on the path towards malignancy.
A true double-edged sword. Hashtag tag tag outro.
Well, this deep dive into hyperplasia has been, I think, a really fantastic look at some of the fundamental rules governing cellular life and tissue adaptation. We've clearly seen its dual nature, haven't we?
Absolutely. On one hand, it's this essential, often life-saving adaptive tool, whether it's regenerating 70% of a liver lobe after donation or preparing the mammary glands for lactation during pregnancy, all powered fundamentally by growth factor signaling and the cell's inherent capacity to divide.
And then we've tracked that very same adaptive mechanism through its potential failure points, the pathologic hyperplasias. Situations where the growth is driven by excessive or inappropriate signals, like too much estrogen flooding the endometrium, or too much DHT stimulating the prostate, or even external threats like those HPV viral proteins deliberately dismantling our cell's regulatory machinery.
Yeah, and the crucial link there, the ultimate takeaway perhaps, is understanding that tipping point, that sustained excessive proliferation, even while it's still technically controlled by the faulty stimulus, acts as a kind of genomic instability multiplier. It just dramatically raises the statistical likelihood that somewhere within that rapidly dividing population, a cell will acquire the specific fatal combination of mutations needed to break free, to achieve independence from external control and become a full-blown malignancy.
Hyperplasia really demonstrates that sometimes the body's own attempt to aggressively adapt and survive by simply making more of itself can paradoxically become the very first step down that dangerous road toward cancer.
It's a profound biological balancing act.
It is. And we know that the balance of hormones and growth factors must be incredibly finely tuned to manage things like that 8-fold increase in bone marrow production we talked about without things spiraling out of control. So maybe here's a final provocative thought for you, the listener, to mull over. When we look at these pathological hyperplasias today, endometrial, prostatic, and others, where do the most pressing research questions lie now? Are we primarily focused on finding better ways to simply overwhelm or silence the excessive presence of the known stimulus, like blocking estrogen or DHT?
Or is there also a significant focus on identifying perhaps more subtle failures, like maybe identifying novel inhibitory factors or pathways that are supposed to act as brakes but have somehow been lost or silenced in these conditions, allowing the normal stimuli to have an exaggerated effect? Where does that critical control failure most often originate? Too much gas pedal or a broken brake pedal?
Yeah, thinking about whether the ultimate solution lies more in restoring a missing brake system versus just cutting the fuel line completely. Well, that probably gives us some real insight into the future directions for both regenerative medicine and cancer prevention, doesn't it?
Definitely food for thought.
Indeed. Well, thank you for joining us on this deep dive. We'll see you next time.